Sarah, go along.
Okay. But guys, I don't like this game anymore. Let's go inside.
Yeah, I got an idea. Let's play some video games.
Yeah. Oh my god. Oh, Jay, this game's awesome.
I know.
Oh, what's it called? I want to get this.
It's called Biosphere.
What Biosphere?
Yeah, it's educational.
Don't give me that educational. What the hell are you making? What? You play educational game? Huh? You think I want to learn stuff?
Educational.
No, really guys, it's awesome. Watch this.
You first, man. Yeah. Not me.
Okay, I'll do it.
You don't see no back man jumping home.
I'm gonna go in.
Are you sure about this? Well.
Level 1.
What's for breakfast?
What the? Where are we?
Where the hell are we?
What the hell? What the hell is this?
Welcome to level 1 of Biosphere. You've entered a time where life hasn't begun yet. Feel free to explore.
Scientists have discovered through these fields that the Earth before life was filled with organic molecules. That is, molecules with a carbon base. These molecules act as the foundation of life. Astronomers have found these carbon-based molecules to be quite common, such as in meteorites, stars, and even other planets. Early life couldn't have been as complex as the modern organisms of today. Life must have been extremely simple. Protein mechanics and the synthesis we know today could not have existed. As we know, the semi-permeable membrane of modern cells are very good at their job. These phospholipids are practically impenetrable. Only with the existence of proteins are they able to move substances in and out of the cell. But life didn't have to start with such complex chemicals. Here's a picture of some simple fatty acids. Not life, but these acids give us a good idea of the possibilities that could occur. Through dispersion forces, these acids can clump together to form a stable vesicle, or in other words, the first cell. This diagram demonstrates that this phospholipid bilayer isn't totally impermeable like the cells we're used to. This is how the cell gathers its required substances. Here the cell continues to grow by attracting more fatty acids through dispersion forces. All it has to do is move, like a magnet. If you move it in range of a metal, they are instantly attracted. This is the fun part. Once the cell grows too big, whatever is moving it, such as waves or rocks or vents, the cell will break. Thus, the surface area to volume ratio is born. Smaller cells stick together, therefore forming the first body. Spontaneous division. So what about genetic material? DNA is highly complex and didn't even exist at this time. All that existed were nucleotides made in the cells earlier. A recent experiment by the University of Harvard has demonstrated that some of these nucleotides are capable of self-polymerization. Monomers will base pair with a single-stranded template and self-ligate. They do this by the simple polar forces which they bond. They can polymerize in solution and spontaneously form new templates or extend existing templates. This forms polymers. But how does all of this become life? Here's how. Our fatty acid vesicles are permeable to nucleotide monomers, but not polymers. Once spontaneous polymerization occurs within the vesicle, the polymer is trapped. Floating through the ocean, the polymer-containing vesicles will encounter convection currents, such as those set up by hydrothermal vents. High temperatures will separate the polymer strands and increase the membrane's permeability to monomers. Once the temperature cools, spontaneous polymerization can occur, and the cycle repeats. The polymer, due to the surrounding ions, will increase the osmotic pressure within the vesicle, stretching its membrane. A vesicle with more polymer, through the simple thermodynamics, will steal lipids from a vesicle with less polymer. This is the origin of competition. They eat each other. A vesicle that contains a polymer that can replicate faster will grow and divide faster, eventually dominating the population. Monomers diffuse into a fatty acid vesicle. Monomers spontaneously polymerize and copy any template. Heat separates strands, increases membrane permeability to monomers. Polymer backbones attract ions, increasing osmotic pressure. Pressure on the membrane drives its growth at the expense of nearby vesicles containing less polymer. Vesicles grow into tubular structures. Mechanical forces cause vesicles to divide. Daughter vesicles inherit polymers from the parent vesicle. Polymer sequences that replicate faster will dominate the population, thus beginning evolution.
What is this? It's like water. I'm so thirsty. It's got green stuff in it. Yes! Yeah! Woo! Yeah! Yeah!